Hey guys, welcome back. We began our exploration in catabolism with the goal to identify where and how our metabolic energy is generated. Yet to this point, we've only generated 4 ATPs, and those ATP molecules have been generated as a substrate level phosphorylation event, meaning that they come almost directly from the breakage of covalent bonds and the energy stored in those covalent bonds that hold the carbon molecules together in the glucose molecule. And none of that initial oxygen has been used in any of these reactions. However, there is no remnants of the glucose molecule left because all the covalent bonds have been destroyed, and the carbons associated with the glucose molecule have been released as carbon dioxide molecules, with the oxygen associated with the carbon dioxide molecules coming from the glucose molecule, not from the oxygen that we see in the aerobic cell respiration equation. So where does the remaining ATP molecules come from? Well, the answer is it comes from the electron transport chain in conjunction with the oxidative phosphorylation reactions. Now, before we look at the physiology, let's first take a look at the four complexes associated with the electron transport chain, and then we'll look at the energy coupling mechanisms that we call chemoosmosis, which is a component of the oxidative phosphorylation. We'll culminate by taking a look at how we can convert the high energy electrons transported by NADH and FADH molecules to the electron transport chain into ATP molecules by using a pump mechanism. Are you ready? Hope so, because here we go. Let's take a look at the structures associated with the electron transport chain that you see here. They're going to be these complexes, Roman numeral 1 through 4. First, take a look and see that 1, 3, and 4 are going to be complexes that are embedded integral proteins. They're going to be transmembranal proteins that will create pores, channels, and these channels are going to be specific to hydrogen ions. Notice, though, also that complex 2 is going to be a hydrophilic complex that's going to be found in the matrix, and it's going to associate with a carrier molecule known as Q. Q's also got a name to it, ubiquinone or CoQ. Now it's important to realize that when these electrons come in, they're going to be transferred from one of these complexes to another. So if electrons come in, as in the NADH molecule, and are unloaded at the complex 1, then they'll be transferred to Q, and they'll be from Q they'll be transferred to complex 3, and from complex 3 they'll be transferred to cytochrome C, and from cytochrome C they'll be transferred to the complex 4. Notice that as these electrons are being transported from one complex to the other, a portion of their energy will be removed, and that energy will be used to drive those hydrogens that is being pumped across those pores. Now note here from the image that you can see that the electrons from NADH are going to be unloaded at the complex 1. The electrons from the FADH are going to be unloaded at complex 2. This means that complex 1 and the pumping that occurs in complex 1 will be driven only by high energy electrons associated with NADH molecules. Now let's look at those complexes a little closer. These complexes are mostly composed of a protein-based series of electron carrier molecules collectively called cytochromes. Now the role of the complexes as well as the cytochromes within them is to gradually remove the potential energy stored in the high energy electrons that they receive from the NADH and FADH molecules, and use this energy to pump hydrogen ions into the intermembranal space. Thus, the electrons and the energy associated with these electrons is used not to generate ATP, but to pump hydrogen ions that will set the stage for ATP production. Anytime we use the word pumping, remember we're talking about active transport. Note that the electrons carried in NADH molecules offload upstream of the FADH molecules. Only the NADH electrons stimulate the hydrogen pumping that is realized in complex 1. For this reason, we will see that the NADH electrons will result in an overall greater production of ATP than we will realize from an FADH electron once we see the culmination occurring at the oxidative phosphorylation. Now, each time the electrons are transferred from one complex to the next, a portion of the energy is drained from the electron. At the end of complex 4, note that the electron is returned back to its resting state. It has no net potential energy left. And these electrons are going to be donated back to a proton in the form of a hydrogen ion. Two such hydrogen ions are going to be covalently bonded to an oxygen molecule to safely store and transport away to the mitochondria. Here is where the oxygen is going to be used. Now here is my best visual attempt to explain this phenomenon in a common sense kind of way. Think of the high energy electrons as being a very energetic baby, screaming baby if you want to think of it. And the protein complexes here as being 4 grandparents. The grandparents each play with the baby. Each time a grandparent passes the baby on to another grandparent, the baby is a little less energized until after the 4th grandparent engages the baby falls asleep and is placed in a safe crib. Except in the case of the protein complexes, the energy drained from the electrons is used to pump hydrogen ions. So we actually take advantage of that energy. Whereas if you think of the grandparent only wished they could acquire the energy that the baby released during that interaction that they had. And note, the crib in this case, for the hydrogen ions is going to be an oxygen molecule to form water, metabolic water.